Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show. Today, bringing another really awesome guest for you involved in science and technology and biotech, creating a better tomorrow for many people out there. Today on the show, we have the honor of being joined by Dr. Jacob Lalezari, who is the Chief Executive Officer of CytoDyn. They are a biopharma company committed to enhancing the lives of patients through really target-specific medicine, and as they say, next generation of monoclonal antibody therapies, and they're specifically focusing on targeting the CCR5 receptor to be used in a platform for a variety of indications. Dr. Lalezari brings over 3 decades of experience to CytoDyn, including 20 years previously with their lead asset, formerly known as Pro-140, and he has served as interim CEO and chief medical officer and has been member of the company's scientific advisory board for several years now. He previously served as the chief executive officer and medical director of Quest Clinical Research, also as the chief medical officer of Virion Therapeutics, and over his career served as principal investigator for numerous phase 1, 2, and 3 clinical studies on a range of viral diseases, including HIV, CMV, HPV, as well as hepatitis, influenza, and RSV, including everything from clinical trials on down. His work has been extensively published, and he is an extremely well-regarded international speaker and patient advocate. Dr. Lalezari received his medical degree around the corner here at Penn, also did his master's at the University of Virginia and his bachelor's at the University of Rochester, and he holds board certification from the American Board of Internal Medicine as well. A lot of really cool themes to get into today. I'm looking forward to this episode. Dr. Jacob Lalezari, thank you so much for taking time to come on the show.
Ira, thank you. That was one of the nicest and most generous introductions I've ever received. To be clear, I became the CEO at CytoDyn in November of 2023, even as I continue to run Quest Clinical Research, which is now in its 36th year of operation, as you said, mostly virology, but also oncology and other indications. So I'm actually doing both at the same time.
Got it. Perfect. Well, yeah, I mean, I'd love to start off, and before we even get to CCR5 and cancer and all that, I just, love to hand you the floor for a couple of minutes just to talk a little bit more about you, because your publication record, I just, I took a glance through it over the last couple of days. I mean, a rather extensive set of work on a range of viral disorders and antiviral drugs, and before HIV even shows up, which we'll get to as well. Tell us a little bit about you. Love to hear your background story, how things started around the corner here at Penn, and then we'll move on from there.
Well, actually, going to medical school was my father's idea. My father was one of the founding fathers of modern immunology, and he was a Persian man, and my mother was a Holocaust survivor. And they met just after the war and came together and wanted a family as quickly as possible. So I was the issue of that marriage of a Holocaust survivor and a founding father of immunology. So if you just extend forward from there, it's little wonder that I ended up spending most of my life working on HIV, a Holocaust of the immune system in gay men. So that's kind of how I ended up there. I'm not sure I actively made a lot of choices. I think I was programmed for it.
Got it. Okay.
But came to San Francisco, and it was to do my medical residency. It was a desperate time. June of 1986, and all these lovely people, young people, were dying terrible deaths with HIV. I helped start the HIV ward at Mount Zion Hospital, and then on June 20th, I had the opportunity, so I went for it, to start a virology research program. And HIV, as you said, CMV, and suddenly there was a giant sucking sound, because suddenly there were drugs that were now becoming available in the world of virology, but there weren't a lot of research institutions that set up to do the clinical testing. So over the decades, as you said, HIV and hepatitis B, hepatitis C, I've really had the privilege of being involved in an awful lot of successful, amazing medical stories, both with the HIV drugs and particularly the hepatitis C drugs, where we were able to demonstrate a 100% cure rate.
Yeah.
So I'm used to success, and bring that attitude to what's happening now with CytoDyn,
Yeah.
because I very much believe in this drug.
Absolutely.
You said Pro 140, we now call it Leronlimab.
Got it. Got it. Yeah, let's talk a little bit about CCR5 or chemokine receptor 5, because it is this really interesting protein that shows up on the surface of immune cells, acts as a receptor for all sorts of chemokines and other biologic moieties that are involved in immune cell migration and inflammatory response. But it is also this important receptor, co-receptor for HIV. And then on previous episodes, a couple of years ago, we profiled, one of the HIV cure cases per the CCR5 delta 32 mutations. So it is a receptor that has a lot of history and a lot of story around it. And I would love for you to sort of take us into that, really from the initial purpose of what it does normally in our body, and then we'll move on in a little bit to how it pops up in various forms of cancer.
Well, you're right. It is fascinating, and it shows up in many chapters of the textbook of internal medicine.
Yeah.
It is one of many receptors on the T cells that is involved with controlling the immune system, moderating inflammation. It sort of creates the GPS system for the movement and migration of cells. So it shows up in infection, inflammation. It's co-opted by cancer. It certainly played a key role in HIV. Its discovery in the mid-90s was because of HIV, where we knew there was a secondary co-receptor that was essential for the virus to get inside the cell. And so they identified CCR5. And then what they did is they looked at it was known that there was a subgroup of the population that had deletion, a 32 base pair deletion in the CCR5 gene that seemed to be related to some historical event that was a selection event. And it's known that, for example, the plague bacillus uses CCR5 to enter the T cell. So it's thought that, particularly in the Northern European population, 10% of the population is heterozygous for this deletion, and 1% of the population is homozygous, and they lack the CCR5 gene and receptor entirely. And when they looked at cohorts of sex workers in the mid-90s who were repeatedly exposed to HIV but remained uninfected, they found out that indeed many of those individuals were CCR5 delta 32 homozygotes, which kind of confirmed the role of CCR5 as a secondary receptor for HIV entry. And so CCR5 blocking drugs are known as attachment inhibitors.
Right.
And so there were a variety of drugs that were developed, both oral drugs and this Pro 140 developed by Paul Madden, which specifically bound to the CCR5 receptor competitively in a way that kept HIV out. And in fact, Quest, my research program in San Francisco, we did the original studies with Pro 140 and showed that it was a pretty good antiviral, that if you administered the drug once a week sub-Q, which is how we continue to do it, you could see about a 1.5 log or 95% decrease in the viral load in HIV positive patients just with this drug. So we knew it was an effective HIV antiviral by 2008 or 9 and published that work. The The problem for Pro 140 is as it was coming to maturity, so too were a variety of other drugs. The integrase inhibitors, Maraviroc, which is an oral CCR5 drug, etravirine, and so nobody quite knew where Pro 140 would fit in to the treatment armamentarium for HIV. So it was kind of pushed to the side and languished until CytoDyn licensed it from Progenics with an intention to develop it as an HIV drug. And indeed, over the years, did complete a phase 3 study showing that Leronlimab works in multi-drug resistant HIV patients. Again, a variety of drugs have been developed that address that MDR population. And at the same time, the drugs for HIV are so good that we hardly ever see a patient now with multi-drug resistant virus.
Right.
So long story, it worked. It was shown to work, but it doesn't really lead to any sort of commercial opportunity or any real benefit for the HIV population, at least right now. But at the same time that was happening, it became clear, as you said, that CCR5 had a much larger role in our lives than just an HIV doorway, and that it was key to inflammation. There are reasons to think it could work in COVID with the migration of inflammatory cells into the lungs. But in particular, it shows up in oncology. And Richard Pastel did a seminal study in mice where he injected a CCR5 positive cell line of triple negative breast cancer into the tails of mice and treated those mice with either a placebo, Leronlimab, or Maraviroc, the oral drug. And he showed that with either of those CCR5 blocking drugs, there was a 98% reduction in the metastatic spread of cancer within those mice. So one of the key roles that CCR5 plays, as I think I said, is like a GPS system for the immune system, directing the traffic and migration of cells. And it turns out that cancer co-opts that CCR5 receptor and uses it as a way of spreading cancer cells and creating metastasis. And that's crucially important because in general, it's not the primary tumor that kills patients, but rather the spread of their metastatic disease. And so to have a drug that, A, has been well tolerated in so many patients, and B, that appears to disrupt the metastatic spread of cancer makes Leronlimab an incredibly interesting candidate.
So again, for our audience, we have an interesting therapeutic that it did well for HIV, but as you said, the tides changed. But nonetheless, you have a very interesting, let's say, repurposing story. We have the co-opting of various types of cancers, breast, prostate, GI, which we'll be getting into because the CCR5 expression, it promotes not just a good migration, but it promotes the migration of cancer cells, the invasion, ultimately metastasis. And that leads us then into the world of, we'll focus on breast cancer initially, but of the triple negative breast cancer diagnosis, because we have, I guess, sort of the three other types of breast cancers that are estrogen receptor positive, progesterone receptor positive, or HEGF receptor 2. So where we have drugs, but those are not the only types of breast cancers. And triple negative is another term for, so this fourth category, and it happens to, a substantial amount of CCR5 expression in these types of cases. Now, I wonder if you could take us down the road and introduce our audience to triple negative breast cancer a little further, and just a little bit of, in terms of the survival and prevalence and so forth of that. I think that'd be really important.
Well, as you said, CCR5 is actually common in a lot of solid tumors, not only breast cancer and all breast cancers, but very common typically in colorectal cancer, most prostate, pancreas, glioblastoma, the solid sarcomas, and the urothelial bladder and kidney cancers. So CCR5 is playing a major role in a lot of important, common, and deadly solid tumors. In particular, as you say, there's this triple negative breast cancer, which unfortunately is the worst of the breast cancers. As you say, it's lacking all the hormone receptors, so estrogen, progesterone, HER2, not present, and therefore the drugs that target those receptors don't work in triple negative breast cancer. And so as a result, triple negative breast cancer occurs in about 10 to 20% of cases. It typically occurs in younger women, often African American or Hispanic, and it often presents with advanced disease. And it's a very aggressive cancer, and a cancer that's responsible for more than its share of morbidity and mortality. And with fairly limited treatment options, the cornerstone of treatment is chemotherapy, both before and after surgery. But as I said, many women present already with metastatic or locally advanced disease. So it's definitely a target for a lot of pharmaceutical companies to try and develop new therapies for this cancer.
And introduce us. I mean, you mentioned, uh, the Pro 140, or as it's known, Leronlimab. It's a humanized monoclonal antibody. It's targeted against the CCR5 receptors. Just say a couple words about the drug, and it looks. I know before, what it was doing in terms of serving as an entry inhibitor for HIV. What exactly is it doing in these particular cases that shuts down this ability of CCR5 to do all these other nasty things that we don't want it doing in these cancers?
Well, it's a great question with an incomplete answer. Unlike the oral drugs, which are allosteric inhibitors that sort of bind in a pocket and then prevent the virus from getting in, leronlimab is a competitive inhibitor
Okay.
that basically completely binds and blocks the receptor. That said, it does not shut down function through the CCR5 receptor the way the allosteric oral molecules do. Instead, it modulates. It's an immune modulator. It
Okay.
basically calms down. It appears to calm down the immune system and lead to more modulated activity. So, for example, the idea was in COVID that it could calm down the rush of CCR5 positive T cells into the lungs in patients in the ICU. And we have some evidence to think that that actually happened. But we think of it as an immunomodulator, even though that word has a little bit of hand waving to it. But the data that we're getting in suggests that however exactly it is acting with that receptor, the clinical benefit is quite striking.
Got it. Got it. Yeah, no, this whole principle of modulating tumor microenvironment and all that, and sort of the interplay between uh, that microenvironment and tumors nowadays is just so prevalent and an important topic. Again, nonetheless, I appreciate you taking us through that. Yeah, I mean, let's talk. I mean, obviously, there's been a lot going on, and most recently was this European Society for Medical Oncology poster that was just presented a couple months ago. This was back in May, long-term survival with Leronlimab treatment in patients metastatic triple-negative breast cancer. Take a little time here, Jay, just to walk through, what's been going on in terms of the clinic, a little bit of what you've seen in terms of survival signals, the phase 2 results, and a little bit of what's happening in terms of, I think, got fast track designation here. A little bit of what the plan is moving forward from where we are now in August.
Absolutely. It is kind of an amazing story. So when I came on as CEO in November of 23, my primary job was to figure out where CytoDyn, should put their resources. There was evidence that it worked in fatty liver. There was an HIV program, an inflammation program. We're doing an Alzheimer's program with Cornell Medical Center in New York. And then there was oncology and the question of whether to do triple negative or colon cancer. And CytoDyn had done 3 clinical studies in oncology, mostly women with triple negative breast cancer, but also other individuals with solid tumors, and enrolled 42 patients across 3 studies during 2020 and 21. And it was right in the teeth of the COVID pandemic. And then, unfortunately, those studies were shut down. Those patients were lost to follow-up. And so one of my primary goals as CEO was to check the box and just make sure what the survival was in that population so that we could publish those results. And it took a while. I just started calling around to the families of those patients, to the investigators at the universities. And I was stunned to find, I remember the first investigator at the University of Arizona emailed me back pretty quickly and said, "Oh, yeah, that patient's still alive. And it's amazing. She has no evidence of disease."
Mm.
And the women who were enrolled in these studies were very advanced patients who had fourth line therapy. They had failed 2 lines in the metastatic setting. About a third of them had brain metastasis. 2/3 of them had organ metastasis. Their survival was based on historical data, should have been measured in weeks and months. And as I'm contacting these investigators at other sites and then calling the family members, some of whom were reluctant to talk to me because they had no clue that CytoDyn had any role in any of this,
Mm.
um, we found patients, actually 8 of the 42 patients from across the cancer studies who are still alive today. And in particular, 5 patients who had metastatic triple negative breast cancer, of which 3 we have recently now confirmed have no evidence of disease, including 2 patients that had lung mets and 1 patient that had lung and brain mets. So it's a retrospective look, it's small numbers, but it's an extraordinary survival story that we were able to, first of all, identify the patients who were alive. And then we went back and requested medical records from all the doctors involved, got those records, pulled them together, put them into a database, and now have shifted it into an electronic database so we can present the whole story to FDA. That has taken months. But the key moment was when we realized who was still alive, and then we paired it with the blood results that we had from the study. And what we found was the common denominator in the survivors was that they were individuals who had been on leronlimab, had induced a protein called PD-L1, which I'll explain in a moment,
Mm-hmm.
to a significant level. And they were individuals who then received a checkpoint inhibitor that specifically blocks the action of PD-L1.
Okay.
And all the women who either didn't induce or induced but didn't get this checkpoint inhibitor, unfortunately, didn't survive. But 100% of the women who did induce and then got a checkpoint inhibitor are alive today. So when a solid tumor begins to grow, it has several challenges. One of them is to create a blood supply so it can grow beyond a 2 millimeter sphere. And the other is to try and keep the immune system from attacking it. And what solid tumors can do, CCR5 positive tumors, is they secrete a protein called RANTES that binds to CCR5 on various cells. And in particular, it binds to a cell called a macrophage, which then flips its polarity. And instead of attacking the cancer, that macrophage now starts helping the cancer.
Mm-hmm.
And in particular, it secretes something called VEGF, which induces the formation of blood vessels so that cancer can now receive blood and nourishment. And then also secreting RANTES, the cancer can attract CCR5 positive suppressor cells. And what those cells do is create that microenvironment that you mentioned that specifically keep the host immune system out and allow that cancer to grow without the immune system recognizing it as foreign and something it should attack. So by using CCR5, it creates a micro or cold microenvironment that allows it to thrive. And what leronlimab appears to be doing is interrupting that signaling between the cancer and CCR5 on the cells and disrupting that microenvironment. Now the macrophages aren't helping the cancer, and in particular, those suppressor cells are not able to keep the immune system from attacking. And as a result, that cancer is now under pressure. And what we saw is patients who received leronlimab, 90% of them induced this protein called PD-L1. And PD-L1 is a protein that cancers can secrete that bind to a receptor on cells of the immune system that are attacking it. And that PD-1 receptor acts as a kind of off switch. So it's a way for the cancer to turn down or downregulate or turn off the attacking immune system. So it is amazing to see that by treating with leronlimab, we are creating a disturbance in that microenvironment, which then causes the cancer to secrete PD-L1 in an effort to turn off the invading immune response. And then we have drugs called immune checkpoint inhibitors that block either PD-L1 or PD-1. And it turned out it didn't matter which one they got. Any patient who got either class of those drugs is alive today. And again, 3 of the 5 have no evidence of disease. So it appeared that there was a second trick up the cancer sleeve, and that just by chance, the patients who got the PD-1 inhibitors, that the cancer didn't have a third trick. And they remain alive and well today, 3 without disease and 2 with apparently stable disease on minimal therapy.
Beautiful. And you know that is not your only recent. I mean, there was another poster session at ESMO, and this was for GI, because you're also doing the colorectal work. And again, here's another, as you were saying, this is another class of tumors that, and you can talk about PMMR and all this stuff. And I'm no expert in the space, but nonetheless, another type of sort of coldish tumor that you need to do things, as you were just mentioning, to sort of wake up and re-educate that microenvironment to kill better and all that. Say a couple words, because I know this was a smaller study, but nonetheless, very, very encouraging sort of phase 1 numbers here that say, hey, this is much more than breast cancer.
Yeah. So to be clear, cold is really a reference to the fact that the immune system is not getting into the microenvironment. When the cells do penetrate, that's when it becomes hot and inflamed. And the PD-L1 expression is a result of that immune invasion. It itself is not what makes the tumor hot. So there were 5 patients with colorectal cancer who were treated in these previous studies. Unfortunately, blood was not collected and PD-L1 was not evaluated in any of those patients. But we saw is that 3 of the 5 had at least a partial response to leronlimab, and 1 of the 5 is also, again, alive today, 5 years later, no evidence of disease. So we have some very provocative signals from small groups of patients that are retrospective in 2 different types of solid tumors, where it appears leronlimab on its own is having some activity as a cancer therapy. And we should point out that leronlimab has been given to 1,600 patients or so. And it's been very well tolerated and is not associated with any sort of dominant safety or toxicity issue, which is not something you can really say about any other cancer drug. That we're seeing a signal retrospectively that the drug appears to be working in both triple-negative breast cancer and in colorectal cancer, and in particular in the patients who happen to get a checkpoint inhibitor. We're seeing evidence of sustained remission, which is, just unheard of in a patient with lung and brain mets. So the onus on CytoDyn now, and the reason I feel enormous pressure every day is that we have to prospectively confirm this. Prospectively confirm both that leronlimab is disrupting the microenvironment and causing this PD-L1 induction, and that providing patients with a checkpoint inhibitor then provides them with significant clinical benefit. If we do that, it's a game changer.
Absolutely. Jason, I mentioned you have fast track designation per the triple negative work at FDA. Could you take us a little bit what's happens next? Because clearly, you're on the precipice here of some major things, and then the same for the GI work as well.
Yeah, I think precipice is the right word. It did take an ungodly amount of time to collate the data from these oncology patients to get the charts from the doctors, because that's become almost impossible anyway. But to get those charts, get the data, put it into a spreadsheet, convert it into electronic database so that we can now have a briefing book that summarizes everything for the FDA. That has taken months for CytoDyn to do, but it is now done. In the meantime, we've started to enroll our colorectal cancer study, have a bunch of sites actively enrolling now. And what we're going to be doing shortly is submitting a request to the FDA for a meeting, at which point we're going to give them a rollover protocol for the colorectal cancer patients that will, we're monitoring their PD-L1 status during the study. And in the rollover protocol, we hope to then provide them a checkpoint inhibitor to see if we can replicate that clinical benefit, that survival benefit that we saw in the breast cancer patients. At the same time, we're submitting a phase 2 follow-up protocol for the FDA in triple-negative breast cancer. And then thirdly, we're going to be submitting a compassionate use protocol for triple-negative breast cancer patients who are otherwise ineligible for our phase 2 study. And in that program as well, we'll be able to verify the induction of PD-L1 and then help those patients where we can to add in a checkpoint inhibitor. So we have the parent CRC study, a rollover with checkpoints for the CRC study, a phase 2 program for triple-negative breast cancer, a compassionate use program for triple negatives, as well. And sort of the third leg of our oncology program is we've been dealing with a couple of key opinion leaders, neuro-oncologists, who have proposed an investigator-initiated study in glioblastoma.
Okay.
And glioblastoma is one of those cancers that uses CCR5. And when you put glioblastoma, co-culture it with leronlimab, those cells express PD-L1. So what they're proposing is we know leronlimab gets into the central nervous system. It gets ferried across, presumably bound to T cells. And we know this from studies in macaques. And the idea from these investigators is we do not know the site of action for the PD-1 inhibitors. We don't know if it has to be in the tumor microenvironment or within the brain, or whether it could potentially be in the periphery. So the idea is with patients with recurrent glioblastoma who have absolutely no treatment options, we want to give them leronlimab in advance of their surgery, measure their PD-L1 induction, and then offer them a checkpoint inhibitor with the hopes that the checkpoint activity is in the periphery, and then activated cells can then enter the brain and attack a cancer that has had a disrupted microenvironment. It's a bit of a bank shot, but one tries what one can.
Yeah,
So
no, we've done a few glioblastoma episodes, and I take my hat off to you if I had it on. I mean that you would, not that there's one that's worse than the other, but glioblastoma clearly falls into that,
right.
class, and so it's great to see that you're thinking about that as well with
Well,
all
the
the different patients.
primary focus, of course, is on breast, triple negative breast cancer, and colon cancer. Um, and then through our EIND program, we'll continue to accept patients with pancreatic cancer, prostate, sarcoma, the urothelial cancers. And in that program as well, we're now able to monitor for the induction of PD-L1. So we're all in oncology. We're all in on this. I believe that leronlimab is showing evidence that it works as a standalone agent. The safety data is so exciting. But this idea that we potentially are offering patients a pathway to a sustained remission is our focus.
Absolutely. Absolutely. Yeah. And again, it's nice coming back to I say it's a very elegant story, but it's also this elegant repurposing story. That's something that, has a few decades of history behind it in one area. And then ultimately you can repurpose for something like this, and, make impact in patients' lives is extremely important. So that's really great. Jay, while we have you, anything else coming up that sort of public facing that we should know about? I mean, obviously, we'll put the link to the CytoDyn website and the pipeline in the show notes, and everyone can go take a look at all the other potential beyond even these bases. But what else is coming up as we get close to 2026 that while we have you
Sure.
today?
Well, in addition to all the interactions with the FDA that are pending, we do have an abstract into the AACR conference in which we'll be presenting details around this PDL1 story. We have an abstract into the San Antonio Cancer Symposium, in which we'll be doing an update on the patients with TNBC. And then at ASCO next summer, we'll be sharing the 5-year survival in those patients. In addition to the work that we've described in breast, colon, and glio, we've been working with Cornell, as I mentioned earlier, to launch a study in Alzheimer's disease. That group now has both IRB and FDA approval and is finally set to start screening patients with mild to moderate Alzheimer's disease. There's a whole lot of reasons why CCR5 is implicated in the pathogenesis of Alzheimer's disease. As well, you had mentioned the cure of an HIV positive individual during a stem cell transplant with a CCR5 negative donor. Well, there's evidence that you can actually use Leronlimab in lieu of finding a CCR5 negative donor. And that work has been successfully done at OHSU by Jonah Sasha in macaques. And so Jonah is now completing a protocol called LATCH, which will try to replicate that cure. But instead of finding the CCR5 donor, we'll be using leronlimab for 6 months to protect the donor immune system from getting infected by HIV, while the graft versus host disease clears the virus out of the reservoir. And so that study is also going to be launching soon. That's very exciting. And then we continue to do some preclinical work, particularly in stroke, where CCR5 seems to play a major role in the response to a cerebrovascular accident where neurons are deprived of oxygen. CCR5 levels shoot up 10,000-fold and shut down neuronal activity and seem to interfere with recovery. And there's evidence in mice that blocking CCR5 can actually expedite recovery from stroke. So we're taking a look at that. All of that on the side while we stay laser focused on our oncology program.
Sure. No, but nonetheless, I'm going to continue to follow, obviously, everything you do in oncology. But yeah, that's I did take a look at some of those papers with CCR5 and neurodegeneration and all that. And hey, I mean, you could have a bunch of blockbusters in here all rolled into one. So it's I'll keep an eye on that one too. It'll be interesting to see how that unfolds in the coming years. But yeah, no, this is an exciting portfolio. It's an exciting background story and how it's evolved over the years. And I wish you and the team the best with it as you continue to develop these, especially the breast cancer indication as it gets close to registration. But again, for everybody out there that's going to be listening to this episode of the show across the various podcast networks, or who will be watching on the YouTube channel, you've been spending time with Dr. Jacob Lalezari, Chief Executive Officer, CytoDyn, doing really amazing things to improve the lives of patients through, as they say, targeted specific medicine in this next generation of monoclonal antibodies. Jay, I want to thank you for taking the time out to walk us through the story and everything you're up to. Obviously, thank you for everything you're doing for all these patients. As we like to say on our show, thanks for helping to create a better tomorrow for everybody. Really an awesome story, and I look forward to continue to follow it.
I appreciate the opportunity. I also want to just acknowledge the investors in our company who have kept faith and shown an enormous patience while CytoDyn rights this ship and heads to open water. So
absolutely. Great having you, Jay.